You picture the ocean floor as barren, pitch black, cold, dead. You're told food chains start with sunlight, plants make energy, herbivores eat plants, predators eat herbivores. Simple, linear, logical. But 2,000 meters down, the sun never reaches. No plants, no photosynthesis. Yet life thrives. Giant tube worms taller than a man, jellies that hunt like sharks, squids that eat the dead. Bacteria that power entire ecosystems without a single ray of light. The deep sea food chain breaks every rule you learned in school. And the deeper we look, the stranger it gets. What happens when the foundation of life isn't sunshine, but chemicals seeping from Earth's core? When whales become islands for hundreds of species? When predators use living light made by hitchhiking bacteria? The deep ocean covers 71% of our planet. It contains 99% of the biosphere, and its food web looks nothing like anything on land. 2022 studying food web dynamics in Monterey Bay's Central Coast Canyon systems, and from research published January 2025 in Ecography Journal analyzing Southern Ocean deep sea food web structure at South Sandwich Islands during toothfish fishing seasons from 2020 through 2022, showing 5 distinct trophic levels with benthopelagic coupling happening primarily between 3rd and 4th levels. Now back to the information. No gut, no digestive system at all, and yet they're thriving in water that should be poison. The temperature swings from 2 degrees Celsius to 30 in seconds. Hydrogen sulfide pours from the vents in concentrations that would kill a human in minutes. The pressure could crush a car. And these worms are growing faster than almost any invertebrate on Earth, 85 centimeters per year in some colonies. How do you eat when you have no way to eat? Oxygen too. And through chemosynthesis, they build organic molecules, sugar, food, energy, not from sunlight, from the chemicals bleeding out of Earth's mantle. The worm provides the bacteria shelter in its trophosome and access to its specialized hemoglobin that can bind both oxygen and sulfide simultaneously. The bacteria provide the worm all its nutrition. The worm absorbs nutrients through the trophosome wall directly into its bloodstream. It doesn't need a mouth. The bacteria are its stomach, its farm, its entire food source. And then bacteria from the environment, free living in the vent fluid, infect the larvae through their skin, like catching a beneficial disease. The bacteria enter through the skin, colonize the midgut, and the larvae's body remodels itself. The trophosome swells, the gut disappears, the mouth vanishes, the worm seals itself into its chitin tube and begins to grow. And the food chain that starts here, powered by volcanic chemistry instead of sunlight, supports 43 different species in the North Pacific alone. 12,490 organisms feeding directly or indirectly on chemosynthetic energy. A study from the Gorda Ridge, 200 kilometers off southern Oregon, found that protists, tiny single-celled predators, consume 28 to 62% of the daily bacterial and archaeal biomass in discharging vent fluids. They're grazing on the chemosynthetic base of the food web and transferring up to 22% of the fixed carbon up to larger organisms. The chemosynthetic microbes at these vents belong to several groups. Campylobacter dominates sulfide-rich basalt-hosted fields where sulfide and thiosulfate oxidation provide the greatest energy yields. The Super5 clade dominates bacterioplankton in highly diluted mixtures of vent fluid and seawater. Sulfur-oxidizing aquaficae occupy narrow thermophilic niches, and some of these bacteria can survive at 113 degrees Celsius. Pyrolobus fumari grows optimally at 106 degrees. Its name means fire lobe of the chimney. Degrees. They host their own bacteria. Everything here either contains symbiotic bacteria or eats organisms that do. Then come the mobile predators, vent crabs, zorsid fish, octopuses. They feed on the grazers and filter feeders, and above them, occasional visitors. Deep diving predators like grenadiers and Antarctic toothfish that venture close enough to snatch prey from the edges of the vent community. The entire structure, 5 trophic levels in some locations, built on chemistry instead of light. An oasis in the desert. The first wave arrives within hours. Mobile scavengers, sleeper sharks, hagfish, rat tails. Giant isopods like deep sea pillbugs. They strip the soft tissue in weeks to 18 months depending on depth and temperature. A whale can lose tons of flesh in this stage. The scavengers come from far and wide, drawn by scent plumes that spread through the water for kilometers. When the flesh is gone, the enrichment opportunist stage begins. Smaller organisms, polychaete worms, crustaceans, colonize the bones and surrounding sediment, feeding on scraps and organic matter leaching from the carcass for several months to 5 years. As anaerobic bacteria break down these lipids deep inside the bone matrix, they release hydrogen sulfide, the same chemical that powers hydrothermal vents. And suddenly, the whale skeleton becomes its own chemosynthetic ecosystem. Osedax worms, zombie worms, bone-eating worms, drill into the bones using acid secretions. They have no mouth, no gut, just symbiotic bacteria in a root-like structure that penetrates the bone and digests collagen and fat. The worms absorb nutrients through this bacteria-filled organ. They can strip a skeleton over 50 to 100 years. Alongside them, vesicomiid clams and bathymodiolin mussels, the same families found at hydrothermal vents, colonize the bones. They harbor chemosynthetic bacteria in their gills. The bacteria oxidize sulfide. The clams and mussels get food. Before whale falls were studied, these clams were only known from cold seeps and vents. Now we know whale skeletons act as stepping stones, allowing vent-adapted species to spread across the abyssal plain. Bay National Marine Sanctuary, they estimated it fell 4 months prior based on minimal flesh remaining and the presence of crabs, eelpouts, and octopuses. That same carcass is likely still feeding organisms today, 6 years later. The whale fall goes through succession, just like a forest after a fire. Different species dominate at different stages. By the time you reach the reef stage, when only mineral bones remain, suspension feeders colonize the hard substrate. But whether this stage always occurs is still debated. Some whale falls disintegrate before reaching it. Others last so long that sediment buries them before the final colonizers arrive. Uptake in the deep sea. Food falls, whales, large fish, squid carcasses contribute somewhere between 0.3 and 4% of total carbon flux to the deep ocean. That might not sound like much, but new evidence suggests it's larger than originally proposed, especially in productive coastal areas where large animals concentrate. Whales feed near the surface on zooplankton and krill. Then when they die, they export that carbon directly to depth. No intermediate steps, no gradual decay, just a massive pulse of energy delivered to one spot. 4 sharks were found on the Angola continental margin over just 1.48 square kilometers of seafloor. If elasmobranch carcasses are that common, imagine how many whale falls are scattered across the 60% of Earth's surface covered by deep ocean. Most of it is abyssal plain, flat, muddy, cold, and the only food that reaches it is marine snow. Think of it as ocean dandruff, continuous shower of organic debris falling from the sunlit surface, dead phytoplankton, fecal pellets from krill and copepods, molted exoskeletons, mucus from gelatinous zooplankton, bacteria, cyanobacteria, all clumping together as they sink. Marine snow forms when phytoplankton in the euphotic zone, the top 100 meters where sunlight penetrates, produce particulate organic carbon through photosynthesis. When they die, or when zooplankton eat them and produce fecal pellets, the material begins to sink. As it falls, particles collide and stick together. Proteins and amino acids act like glue. Transparent exopolymer particles from phytoplankton exudation help aggregation. The flakes grow. Some reach several centimeters in diameter. Population lived despite the possibility of horizontal displacement by currents. Salp fecal pellets sink even faster, over 1,000 meters per day. Transit time to the deep is days to weeks. This creates a temporal coupling between surface production and seafloor sedimentation. When phytoplankton bloom at the surface, the deep sea gets a food pulse weeks later, seasonal, predictable, like leaves falling in autumn. But most marine snow doesn't make it to the bottom. Between 1 and 40% of primary production is exported out of the euphotic zone. That export flux attenuates exponentially through the mesopelagic zone, 200 to 1,000 meters depth. Only about 1% of surface production reaches the seafloor. The rest is consumed or decomposed in the water column. Bacteria colonize the particles. A 25-day study in the Northeast Atlantic found that bacteria and cyanobacteria on marine snow aggregates were 100 to 6,000 times more concentrated than free living populations, depending on depth. Cyanobacteria concentrations were 3,000 to 2,500,000 times higher on aggregates. The aggregates are hotspots for microbial activity. Protists, copepods, amphipods graze on the bacteria. Larger animals eat the grazers. They're not just floating bags of water, they're active hunters. And many deep-sea animals feed on marine snow aggregates. The vampire squid, Vampiroteuthis infernalis, was thought to be a fierce predator based on its name alone. Vampire squid from hell. Dark red body, huge blue eyes, proportionally the largest of any animal relative to body size. Cloak like webbing between 8 arms, rows of cirri that look like teeth. But in 2012, researchers at Monterey Bay Aquarium Research Institute finally figured out what it eats after a century of mystery: marine snow. The squid extends its filaments like fishing lines and lets marine snow accumulate on them. Dead bodies of microscopic algae, fecal pellets from copepods and krill, mucus debris from larvaceans that build feeding filters. When enough material collects, the squid retracts the filament, scrapes off the marine snow with its arms, coats the particles in mucus to form food balls, and transfers them to its mouth. It only needs to feed about twice per week. Its body is neutrally buoyant, so it doesn't expend energy staying at depth. It doesn't swim to find food, it just waits. This energy-efficient lifestyle allows it to survive in one of the most nutrient-poor environments on Earth. Lifetime. Lifespan potentially up to 10 years. They don't reproduce in massive single clutches. They spread reproduction across years. This strategy works in the deep sea where food is scarce but predation is low. The vampire squid represents a fundamentally different approach to the cephalopod life cycle. Slow, patient, opportunistic. It feeds on death, on feces, on snot, on the biological refuse sinking from above, and it thrives. Station M Time Series in the California Current has been monitoring carbon export at 4,000 meters depth for 3 decades. The data shows carbon export has been increasing. DNA sequencing and microscopy of archived sediment trap samples are revealing why. The composition of marine snow is changing. The organisms involved are changing. Climate shifts in surface productivity are lengthening food webs in the deep sea. A productivity hypothesis from 1927 suggested that years with higher primary production have longer food webs. The Southern Ocean data confirms it. Food chain length at South Sandwich Islands varied across 2020, 2021, and 2022 fishing seasons based on net primary production levels. More productivity at the surface means more trophic levels in the deep. The ecosystem responds, expands, contracts, based on what falls from above. The deep-sea serratioid anglerfishes are the most extreme. They live in the bathypelagic zone below 1,000 meters where sunlight never penetrates. The females have a modified dorsal fin spine called an elysium that extends like a fishing rod from their forehead. At the tip is a fleshy bulb called an esca packed with bioluminescent bacteria. The bacteria are environmentally acquired. Anglerfish larvae hatch without symbionts. Female larvae develop a pore in the esca as they mature. When the pore opens and contacts seawater, bacteria colonize it, specifically bacteria from the genus Vibrianaceae, with extremely reduced genomes adapted to a symbiotic lifestyle. Only one other symbiont species was found, with a specific relationship to Cryptopteros quasii. Host and symbiont phylogenies lacked congruence, no statistical support for codivergence. This means the bacteria aren't passed from mother to offspring. They're picked up from the environment each generation. Somehow these bacteria with severely reduced genomes, bacteria that are obligate symbionts dependent on the host, maintain free living. Populations in the deep ocean. They survive in cold, high-pressure, low-nutrient water. And when anglerfish larvae drift by and present the right habitat, the bacteria colonize. Researchers even recovered symbiont-specific gene sequences from water collected near anglerfish hosts. The bacteria are out there, floating, waiting. It moves the elysium back and forth with 5 distinct pairs of muscles. Erector and depressor muscles move the elysial bone. Inclinator, protractor, and retractor muscles move the pterygoiophore. The esca emits blue light that water absorbs least. Small fish, crustaceans, anything curious about the light swims close. The anglerfish waits, drifts, doesn't actively swim. Ambush predator. When prey is close enough, the anglerfish strikes with suction feeding. Its jaw and stomach can distend to consume prey up to twice its size. Bones are thin and flexible. The jaw extends all around the anterior circumference of the head. Bands of inwardly inclined teeth line both jaws. The teeth can be depressed to let prey glide in but prevent escape. This feeding strategy is perfectly suited to the energy-poor deep sea environment. Don't waste energy searching. Let the prey come to you. And they stole this ability from bacteria through the food chain. Dragonfish eat copepods. Copepods eat far red light absorbing bacteria. The dragonfish acquire the bacterial chlorophyll through their diet and use it to hunt with their own private wavelength. They see prey. Prey doesn't see them. The cookie cutter shark uses bioluminescence as camouflage and lure. It has thousands of tiny photophores on its ventral side, emitting greenish counter-illumination light. From below, it blends with downwelling light from the surface, invisible, but it leaves a small, darkly pigmented patch under its jaw unpigmented. This dark spot mimics a small fish. Larger predators see what looks like prey and approach. Then the cookie cutter shark takes a circular bite from the larger animal. Elephant seals, whales, tuna. All found with cookie cutter scars. The arm keeps glowing from inside the predator's stomach like a burglar alarm, attracting larger predators to the scene. Some animals use bioluminescence for counter-illumination. Photophores on their ventral surface match dim light from above, making them harder to see from below. The hatchetfish is famous for this. Flat, reflective. Photophores precisely calibrated to ambient light levels, nearly invisible when viewed from beneath. And bioluminescence isn't just for predation, it's for communication, finding mates in the vastness. Male Caribbean ostracods use bioluminescent signals on their upper lips to attract females. Psyllid fireworms move to open water during the full moon, where females use bioluminescence to attract males while swimming in circles. Even anglerfish use light for mate recognition. The sexual dimorphism in anglerfish is extreme. Males may be several orders of magnitude smaller than females in mass. In some species, the male becomes a sexual parasite. He finds a female, bites into her side, fuses with her tissue. His circulatory system merges with hers. He becomes a permanent sperm-producing appendage. Multiple males can attach to one female. The male loses his eyes, fins, most internal organs. He's reduced to gonads and a basic life support system fed by the female's blood. When the female is ready to spawn, the attached males release sperm. This solves the problem of finding a mate in an environment where you might encounter another individual once in a lifetime. This is where most of the ocean's biomass lives, not at the surface, not on the bottom, in the middle. And a November 2024 study published in Nature Communications found that deep sea mining discharge could disrupt this entire zone. Researchers at University of Hawaii studied the Clarion-Clipperton Zone in the Pacific, where companies want to mine manganese nodules. The mining process pumps sediment and waste up into the midwater, creating plumes of particles. The study found that 53% of zooplankton and 60% of micronectin, small swimming organisms, would be affected by mining waste discharge. The particles are like junk food. They contain far fewer amino acids than natural organic matter. Major source of nutrition gone. And the effects cascade up. Micronectin are eaten by larger fish. Larger fish are eaten by tuna. Tuna are caught by commercial fisheries. The deep seafood web connects directly to human food security. Third level was cephalopods and crustaceans. The benthopelagic coupling happens primarily between third and fourth levels. Pelagic squid and crustaceans, including euphausiids, make up the third level. Demersal fish, like grenadiers, Macrurus species, occupy the fourth level. Stomach content analysis elsewhere in the Southern Ocean confirms squid and crustaceans dominate grenadier diets. These organisms perform diel vertical migration. They rise to feed at night, descend during day. They're a nutrient pump. Energy from surface water is transported to the deep, and predators like toothfish and grenadiers connect pelagic and benthic realms through predation. Kilograms body weight? It's in the fourth trophic level. But Disostachus mawsoni, the Antarctic toothfish in the fifth level as a top predator, eats colossal squid despite the size difference. Conversely, Moritethus longemana reaches 110 centimeters mantle length but sits in the third trophic level. Smaller species like Chiroteuthis varani at 30 centimeters occupy the fourth level. Traditional predator-prey size ratio models don't work here. The deep sea food web is structurally different from surface ecosystems. Trophic redundancy is high. Multiple species occupy the same functional role. This makes the system resilient to species loss but vulnerable to systematic disruption. The Southern Ocean study found food chain length varies with net primary production. Years with higher productivity have longer food webs. As climate change increases productivity in some regions through nutrient upwelling and stratification changes, deep sea bathyal food webs in slope and seamount areas will lengthen. More trophic levels, more complex energy pathways. But increased productivity also means increased respiration by heterotrophic microbes and zooplankton in the mesopelagic zone. More carbon recycled before it reaches the deep. The biological pump efficiency changes. And if mining removes the organisms that connect surface production to deep consumption, the whole system destabilizes. MBRA's 27-year ROV video dataset from 1991 to 2016 revealed food web connections no one expected. Jellies eating larvaceans. Larvaceans eating marine snow, but also producing mucus houses that become marine snow when abandoned. Predators eating predators in cycles. Specialist scavengers that only appear at food falls. The food web isn't a chain or even a simple web. It's a three-dimensional network distributed vertically through the water column with temporal dynamics tied to surface productivity cycles and spatial heterogeneity around vents, seeps, and food falls. Submarine canyons act as biodiversity hotspots. They channel nutrients, focus currents, create upwelling. The Monterey Canyon has higher biomass than surrounding areas because it funnels organic matter from coastal zones into the deep. A USGS-led study on deep seafood webs and submarine canyons found that carbon transport, transformation, storage, and sequestration in these systems is vital for regulating Earth's climate. Deep-sea organisms play pivotal roles despite dwelling in complete darkness, dependent on nutrients from the surface. The canyons create corridors, highways for energy, moving from coastal productivity to abyssal depths. And within those corridors, species assemble into communities structured by depth, oxygen levels, temperature, and food availability. The superbenthos and zooplankton at the base of canyon food webs show assemblage patterns that shift with depth and season. Different communities dominate different zones. The food web structure isn't uniform, it's patchy, dynamic, responsive to local conditions. Green sulfur bacteria at hydrothermal vents require both chemical energy from hydrogen sulfide and light energy to survive. But there's no sunlight at vents. Instead, they capture energy from the weak radioactive glow emitted from geothermally heated rock. Chlorosomes in these bacteria are so efficient at harvesting light that they can grow at intensities 1,000 times lower than other photosynthetic organisms. They're photosynthesizing in the dark using geothermal radiation. This was discovered at the first zoanthids, relatives of coral, ever recorded at a hydrothermal vent. Before this, we didn't think photosynthesis of any kind was possible in the deep sea. The discovery completely changed our understanding of energy limits for life. Reflectance less than 0.5%. A study published July 2020 in Current Biology found 16 species across 7 distantly related orders with this adaptation. The skin has a continuous layer of densely packed melanosomes in the exterior-most dermis. No unpigmented gaps between pigment cells. The melanosomes are optimized in size and shape to minimize reflectance. Why? To avoid reflecting bioluminescence. When a predator lights up to search for prey, anything reflective glows. Ultra-black fish absorb the light, invisible even when illuminated. This is biological stealth technology, and it evolved independently multiple times because the selective pressure was so strong. In an environment where light equals death, being blacker than black is survival. It starts with chemistry, with death, with bacteria. It's powered by volcanic vents, whale carcasses, and the slow rain of debris from above. Predators use living light made by bacteria they catch from the environment. Prey become invisible by absorbing that light with melanin structures more efficient than human-made materials. Jellies hunt like sharks, squids eat snot, worms survive without mouths, fish eat food twice their size. Size doesn't predict trophic position. Productivity at the surface controls food web length in the abyss. Mining waste could poison organisms we're only beginning to understand. And after studying the ocean for centuries, we're still discovering fundamental processes. Green sulfur bacteria photosynthesizing with geothermal radiation. Anglerfish bacteria maintaining free living populations, despite being obligate symbionts. Vampire squids living 10 years while their shallow water cousins die at 3. Behaviors we couldn't imagine. A 25-year data set, and we're still finding patterns. Station M, at 4,000 meters, shows carbon flux increasing for 30 years, and we're just starting to understand why. The Southern Ocean toothfish data from 3 fishing seasons reveals food web structures that differ from every other ecosystem. Each study answers questions and raises 10 more. In 2021, researchers found the first whale fall on the mid-Atlantic ridge. They monitored 1 year of ecological succession. The progression matched Pacific observations, but with regional differences. Different species, different timings. The Atlantic deep sea food web has its own rules. In 2019, explorers discovered an active whale fall at 10,000 feet in Monterey Bay during a live stream. Thousands watched in real time as ROVs documented crabs, eel pouts, octopuses feeding on the carcass. That whale is still feeding organisms, still cycling carbon, still supporting dozens of species in an area that would otherwise have almost nothing to eat. And we only found it by accident while studying something else. Millions of organisms documented, and that's one canyon system in one part of one ocean. The dataset represents a tiny fraction of the deep sea, a single point of light in an ocean of darkness, but it's enough to show that everything we thought we knew was wrong. The food chain isn't a chain, it's a 3-dimensional dynamic network with chemosynthetic bases, light-based communication, particle feeding specialists, and size-independent predation. It's built on symbiosis, on bacteria living inside animals providing all their nutrition, on larvae catching infections that save their lives, on dead whales becoming reefs, on feces and mucus feeding a football-sized squid for a decade. On climate patterns, one disruption anywhere cascades through the system. Mine the nodules, and you poison the zooplankton, and you starve the micronectin, and you collapse the fishery. Warm the surface, and you change bloom timing, and you alter marine snow composition, and you shift food web length in the abyss. It's all connected, surface to seafloor, light to dark, life to death, and death back to life through bacteria that make food from poison. The deep sea breaks the rules because it has different rules. When you can't photosynthesize, you chemosynthesize. When you can't see, you make light. When food is scarce, you eat the dead. When finding a mate is nearly impossible, you become a permanent parasite. When predators use bioluminescence, you become blacker than space. Evolution finds a way. And it finds ways we never imagined because we barely looked. 2025. Almost 250 years since the first deep sea samples, and we're still discovering baseline ecology, still finding new phyla, still learning how energy flows through the largest ecosystem on our planet. While dinosaurs rose and fell, the vampire squid floated in the oxygen minimum zone eating death. While continents split and merged, tube worms grew around vents powered by Earth's core. While ice ages came and went, jellies hunted in the twilight zone with bodies 95% water. The deep seafood chain isn't new, we're just new to understanding it, and every answer makes the ocean stranger. In 2024, a sediment core from a deep Pacific site showed whalebone fragments mixed with vent community fossils. Chimney pieces from extinct vents, Osedax burrows and ancient bones. The communities overlap in space and time. They share species. They're not separate ecosystems, they're nodes in the same network. And that network has been operating for millions of years while we walked around on land thinking photosynthesis was the only game in town. It's not. Chemosynthesis might be older, might be more fundamental, might represent how life started before oxygen was even in the atmosphere. The deep sea food chain could be the original food chain. And everything else, plants, sunlight, herbivores, the whole terrestrial web, might be the weird exception, the shallow water variant of a system that started in the dark. In late 2024, researchers analyzing seabed video from the Clarion-Clipperton Zone found something unexpected: active hydrothermal vent communities on the abyssal plain far from any known spreading ridge, isolated, surrounded by nodule fields targeted for mining. The vent communities included species found nowhere else, tube worms and mussels with symbiotic bacteria, but different species from ridge vents, different chemistry. Different evolution. The vent wasn't on a tectonic boundary. It was powered by serpentinization, water reacting with mantle rocks exposed through faulting, producing hydrogen and methane, chemosynthetic bacteria using hydrogen instead of sulfide, an entire parallel vent ecosystem with its own rules. And it was discovered in the middle of a mining lease area. One more dive, and they might have destroyed it before knowing it existed. That's the deep sea, 1% explored, 99% unknown. And the food chains down there are stranger than we have words for. The deep sea food chain isn't simpler than shallow systems. It's vastly more complex, more distributed, more dependent on microbial interactions and symbioses we're only beginning to document. And it's all happening in permanent darkness under crushing pressure in the cold, while we sit here in the light, thinking we understand how life works.